Alright, we're starting Unit 2 of Microbial for the second test in the class. Alright, this starts out with microbial nutrition, definition of metabolism, the sum total of all the chemical reactions that occur in a cell. So that's pretty simple. Catabolic reactions, so these are the ones that break things down, so they release energy. Anabolic, like anabolic steroids, builds things up, and so it requires energy. And yeah, everything we know about metabolism is based off of cell culture. So that means a lot aren't studied. Enzymes, these are catalysts. They reduce the activation energy of chemicals going through reactions by making the reactants more easily get to the transitional state. Not important for this. Typically, they rely on hydrogen bonds, van der Waals forces, and hydrophobic interactions. They also rely on having hydrophilic interactions on the outside, but I don't think we're going that specific. My background is in biochemistry, so if I go off the deep end, you know why. Ribozymes are also a thing, but not important for this test, apparently. Only important thing here is that microbes have an acidic condition around them. That'll come up later when we talk about their version of energy generation, or their version of the electron transport chain. Catalytic site, so has a substrate, binds to the active site, and then function is performed. I don't know what this is about. Enzyme with glycolytic pathway substrate, right? Oh yeah, actions will always move towards equilibrium. That's just a basic chemistry thing. Oh, you can go into NCBI and find once you get the protein sequence, you can introduce random mutagenesis to find what parts are important for function, then you can try and recover said function. Is the basic way to do that experiment. Saccharomyces cerevisiae is used in fermentation. It will make ethyl alcohol, also known as ethanol, which you find in beer. It'll do this after the will it reduce the it'll take pyruvate, it'll change it into some type of aldehyde, and then it will, I suppose it'll reduce it. No. The substrate will be reduced. There we go, and form ethyl alcohol. Also used in making bread as a leavening agent. It's a fungi, so it has nucleus, mitochondria, organelles, and it's larger than bacteria. Can grow as single cells. Can also have a diploid stage where two different strains of it mate. Chitin at cell walls. Yeah, none of this is important. Fun fact, IPAs come from when they would ship beer, I think they're supposed to say, from Britain to India, and they would make it extra hoppy so that it would have an antimicrobial effect, and so that the beer wouldn't spoil. And that was actually safer to drink than water at the time, because sanitation. Alright, Catabolic Diversity, so this is the first list of things. FAACP, so fermentation, anaerobic respiration, aerobic respiration, chemolithotrophy, and chemolithotrophy is it's going to get carbon source of chemicals and then electron source of water, or some inorganic element like water, and then phototrophy is obviously using sunlight. For this test, we're focusing on aerobic and anaerobic respiration and fermentation. So fermentation, anaerobic respiration, aerobic respiration, chemolithotrophy, phototrophy. Alright, so can we repeat it? Fermentation, anaerobic respiration, aerobic respiration, chemolithotrophy, and phototrophy. Alright, on to energetics. So fermentation and respiration. Fermentation, this is going to be substrate level phosphorylation, and you're going to continually cycle. The reducing agent will continually be the final electron acceptor as well, and it's like 2 ATP per cycle. Respiration uses oxidative phosphorylation, and so the ATP is produced from the proton motive force, and that's created by an electron transport chain. And basically the oxygen that comes in to accept the electrons is exogenous. Alright. This is more biochem stuff. We just have to know, or you should just know, that there's a net of 2 ATP, 2 pyruvate, 2 water, and 2 NADH and 2 H plus ions from glycolysis, which is this anaerobic form of respiration. Essentially, fermentation will contain glycolysis plus an extra step. It'll be either NADH reducing pyruvate, or there'll be an extra step in there. Right, we'll get to that. Citric acid cycle. This is completely oxidizing pyruvate, so you get 2 pyruvate out of each cycle of glycolysis, and so you now need to remove all of those carbons and make them CO2 and harvest the energy. We don't have to know in depth, just know that there's 6 CO2 molecules released and 38 ATP generated, counting glycolysis before. So that's the 2 from glycolysis, 2 net from glycolysis, and then 36 net from citric acid cycle. And the electron transport chain, which is where you get the ATP. Alright, and this is a key regulated site in catabolism and biosynthesis and cell signaling and everything else. To ferment or not to ferment is based on oxygen presence in those that can switch between it. Pyruvate is shuttled within mitochondria and eukaryotes, so the glycolysis happens out in the cytoplasm, and then pyruvate moves into the actual mitochondria. And prokaryotes, this all occurs within the cytoplasm because they don't have organelles. And 6 CO2 also produced in bacteria. And intermediates, of course, do other signaling things. Citric acid cycle, we don't have to know everything about it, which I think this is just showing the various functions that can be maintained by the signaling of these citric acid cycle intermediates. Electron transport chain, it is membrane associated. The mediation of electrons from primary donor to terminal acceptor, so it goes through a series of proteins that will pump protons either into the intermembrane space in eukaryotes in the mitochondria, or just outside of the cell membrane in bacteria. I guess prokarya more appropriately. Conserve some of the energy released during transfer and used to synthesize ATP. Okay, that's what I just talked about. And this is where NADH and FADH2 come in, and the various cytochromes and things are those proteins that will pump the protons. And this is step 3 of metabolism. The first being glycolysis, the second being the citric acid cycle, and the third is the electron transport chain. And sometimes the electron transport chain is called the final. You could arguably say that the ATP synthesis is the 4th step. I'm not sure what this particular professor wants us to think. Generation of the proton motive force, so these are the hydrogen moving outside. Then there will be an ATP synthase somewhere where they'll move back down, and that movement down its gradient provides the energy for ATP production. There's 2 points of entry. NADH comes in on complex 1, and FADH2 comes in on complex 2. So NADH has slightly more energy, or I guess it'll pump slightly more hydrogen out than an electron from FADH2. So this is showing substrate level phosphorylation is just from one energy-rich intermediate to a different intermediate. In this case, ATP, and then this is the proton motive force used in oxidative phosphorylation. As you can see, they're quite different chemical schemes, but they both end up with ATP production. Proton motive force. During electron transfer, several protons are released, causing a slight acidification of the external surface. So again, that's the pumping out of the hydrogen, and all of the protons originate here from NADH and the dissociation of water. Hydrogen, yeah. So after it drops off the hydrogen and the electrons, those will eventually go and get picked up. Results in generation of pH gradient in an electrochemical potential across the membrane. So gradient, whenever there's a high concentration of something, it'll want to move to a low concentration, and so by making it higher on one side of the membrane, you make it want to move into the other side, and it can be used to do work. So the outside is going to be positive relative to the inside, which is negative relative to the outside. This is ATP synthase, this is the wheel, so there's lots of hydrogen on this side, it'll move through, and somehow that cranks this wheel and puts another phosphate on ADP to make ATP. It's called the molecular generator because it physically moves like a turbine. And we did not talk about the free radicals. The essentials of fermentation: you have an organic compound of some kind. It is oxidized by NAD+, and it actually turns into some type of energy-rich compound, which then has substrate level phosphorylation of ADP into ATP. Now it's oxidized, and then it will be reduced by NADH and turn into whatever its fermentation product is. So you need to be careful of these fermentation products because they can be lethal to the cell. So this is just more description of fermentation. Enzymatic catabolism, so the breakdown of carbohydrates generate ATP, and it does not use the electron transport chain. It uses an organic molecule as the final electron acceptor, so it can occur in the absence of oxygen. So anaerobes will use fermentation, and there are a lot of types. You can have ethyl or lactic acids, or mussels make lactic acid, and obviously yeast make ethanol. So this is the alcohol fermentation. You have glycolysis, you get your NADH, so this is your organic compound is glucose, and then your energy-rich compound is this pyruvate, and then that turns into acetaldehyde in this version, but that's unimportant. Your fermentation product, finally, is the ethanol after this gets reduced. And so you can also see lactic acid fermentation if your organic what is this called? Organic compound. Then you have your energy-rich compound, so pyruvate is the energy-rich compound. And then you get your oxidized compound. Oh, maybe pyruvate is your oxidized compound. That would make more sense. Alright, so pyruvate is your oxidized compound. And then your fermentation product after getting reduced. So I was wrong the first time. This is the oxidized compound. So pyruvate is always your oxidized compound. Fermentation generates ATP solely through substrate level phosphorylation. Talked about that. First step is fermentation is glycolysis. So you have glycolysis, and then you have the actual fermentation process, I assume, is the turning of pyruvate or acetaldehyde into the fermentation product by reducing it with your eternally regenerated NADH. Differences, obviously, oxidative phosphorylation uses proton motophores, ATP synthase, and that chemiosmotic gradient. ATP as a molecule, this is describing why it has energy. It's because those phosphates are negatively charged and want to move from each other, away from each other, because negatives repel each other, like charge always repels each other. And so there's a lot of potential energy locked into that bond that you break when you break ATP. Fermentation. Yeast and several bacteria carry it out. Lactic acid is by the bacteria that make yogurt and by your muscle cells, which this talks about here, and that's only when oxygen is sufficient. So when you exercise enough, you'll hit that. Respiration has the advantage of producing 19 times more ATP, so it's that 38 instead of just 2. And starch and glucose are hydrolyzed to produce glucose, which is an incorrect sentence because glucose can't be hydrolyzed to produce glucose. But starch is broken down to glucose, and then they're hydrolyzed to make this ATP. This is just a bunch of things that can be broken down anaerobically. It's basically every macromolecule present in the cell. There's a huge diversity of different things that can be fermented. This is just my personal notes for this test. Is the whole section on fermentation was quite emphasized. Alright, exponential growth. Most bacteria have shorter generation times than eukaryotic microbes. E. coli is the best example. It has a doubling time of about 20 to 30 minutes. And so it grows really fast, whereas yeast are 90 minutes or longer. Generation time is dependent on the growth medium and incubation conditions. So whatever medium we put it in, I just put some yeast that are in minimal media, and it triples their doubling time or something ridiculous. So it can be quite a significant impact. Alright, so this disgusting picture is a Streptococcus pyogenes infection, and so streptococci are capable of producing a hemolytic agent, and it does that because it needs iron. That's what hemolytic pathogens are doing. The toxin is called staphylococcal toxin. This is what causes pharyngitis. Its doubling time is 20 to 30 minutes, so the same as E. coli. It is pus forming. I assume it is gram-negative because it causes disease. I'm not sure that's true. It can cause scarlet fever. It's the causative agent, and then this is the strep pattern. It grows in a chain. Causes strep throat, can grow in blood agar, but can grow in other places too. I will have to check and see if this is truly gram-negative. I thought it was until I remembered it was cocci. I'll look into it. Maybe I'll post that after this video. Exponential growth. Growth of a microbial population in which cell numbers double within a specific time interval. So this is basically just telling you that it's going to follow that exponential curve of it's going to look really slow at first, but then all of a sudden it will rapidly increase. It'll eventually plateau, and then it'll start going down for the death phase. Alright, rate of microbial growth, easiest to see with a logarithmic curve. Will be the easiest to figure out the doubling time. And if you look at the actual curve, you can see that exponential growth I was talking about. On this test, basically, I have to find 2 points that are exactly doubled numbers, so in the total cells. or either the total cells or the cells per unit volume, so the concentration, to see what the doubling time is. And that's just a simple matter of looking at the number of hours. Then you might have to divide based on that. Exponential growth, more of it. Geometric progression of the number 2, so that's how you get this equation. This n is your total number of bacteria at the end, not the concentration. Very important. This is the actual final cell number. N sub naught, that is the initial cell number. That's the total cell number, so if you have a concentration, you need to multiply by the volume to get this. And then 2, this is because it's geometric progression of 2. And then N actually stands for generation time over time. And that'll make more sense on the next slide. So, generation time is time over the number of generations. I was a little bit off there because I haven't looked at this in a while. So it does stand for generation time, but generation time is defined as the duration over the number of generations. So duration of exponential growth, so this is going to be different depending on your bacteria, and that's why it needs to be unique to them. And then n is the number of generations, and so that's pretty straightforward. Obviously, as the T goes up, weird things start happening. Alright, typical growth curve for bacterial population. I'm going to need a calculator on this test because of that. Well, in the previous 2 slides, those equations, and maybe I'll make a separate video about them. We'll see if I get to that before the test. There's phases of growth here. First, there's the lag phase. That's the bacteria getting used to whatever you're culturing in them. Then there's the exponential phase. This is the exponential growth that was described before. Then, as resources start to be used up, you reach a stationary phase. During the stationary phase, you see endospore formation of gram-positive bacteria because they try and protect themselves from entering the death phase, which, as resources and toxins build up, they start dropping off. Important to note that the death phase does not have as steep a slope as an exponential phase. Alright, I don't think there's anything important about those. Amoxicillin, the best time to use it is on the exponential phase because it will disrupt. Amoxicillin works by disrupting cell wall synthesis, and so the only time cell walls are being really built all that much is during the exponential phase. And you have the added bonus of avoiding the time when endospores have formed. And it works as a competitive inhibitor, inhibiting transpeptidase, which makes the pentaglycine bridge that was discussed for the previous test. The best time to use is during the exponential phase, that's what I just said. If something is staining negatively that should be staining positively, it's probably in the death stage. So you want to gram stain in the log phase if you want an accurate gram stain. Alright, the microbial growth cycle. Lag phase, just talked about that. That's when it first begins. Exponential, this is the healthiest representation of these bacteria. Stationary phase, their population growth is 0. And usually essential nutrient gone or waste product is accumulated to stop growth or stop division. Measurements of total cell number. There's a couple of different ways to do this. Microscopic counts is the first way. This is hard to distinguish between living and dead. I've actually done this, and it takes a lot of practice before you're able to tell for sure what's living and what's dead. And even if you are pretty confident, Some dead cells, especially if they're recently dead cells, can still take up dyes and stuff, so that could be an issue. Small cells can be difficult to see or can be overlooked. Pretty commonsensical. There's precision is difficult to achieve, and then phase contrast is required if stain is not used. Alright, so the first one, C. So CSPA. I guess can see pretty animals. That seems like a bad. Oh, I could totally go with caps. And so I'm going to go with that. So instead of the order you see here, put it in CAPS order. Caps cannot distinguish between live and dead cells. A phase contrast microscope is needed if stain is not used. Precision is difficult to achieve, and small cells difficult to see and can be overlooked. So cannot distinguish between live and dead. A phase contrast microscope is needed if there's no stain to use. Precision is difficult to achieve. And then S, so caps. Small cells difficult to see and be overlooked. Alright, again, since I didn't get it. Caps cannot distinguish between live and dead. The A is a phase contrast microscope is needed to if no stain is used. P, precision can be difficult to achieve, and then S is something about specific. No, small cells are difficult to see and can be overlooked. Alright, so caps. Cells that are alive and dead are difficult to distinguish, can be difficult to distinguish alive and dead. Phase microscope is needed if there is no stain. Precision can be difficult to achieve, and small cells can be overlooked. Alright, got it. Measurement of total cell numbers. Microscopic counts. Limitations of microscopic counts. Cell suspensions of low density, less than 10 to the 6th cells per mil, so 1,000,000 cells per mil, are hard to count. Motile cells need to be immobilized, and debris in sample can be mistaken for cells. So more limitations. So cell suspension of low density, CMD. Can microscope death. Alright, cells can be suspended at low concentrations. Can microscope death. Can microscope. Motile cells need to be immobilized. I'm going to go with just CMD. Can my microscope. So cells are in a low concentration. Immotile cells need to be immobilized, motile cells need to be immobilized, and debris can be mistaken for cells. So cells need to be at are in a relatively low concentration. Mobile cells need to be immobilized, and the debris can be mistaken. Direct microscopic counting procedures. So this is a cytometer, not to be confused with a hemocytometer, where you do direct cell counts over an area. Not commonly used. Alright, viable cell counting. So this would be like plating things. So the two major types of plating are the spread plate and the pore plate. The spread plate is basically you spread the bacteria over the top, and the pore plate is you literally pour the bacteria into the media before it's settled. And these are nice because you will only count colonies that originated from one unique, definitely alive cell. Because if it was dead, it could not give off progeny. And these are more commonly used than, say, the cytometer. And you do it by serial dilution to make sure that you do not have too many cells or too little cells. they should be in the 20 to 200 range number of cells on the plate to be an appropriate viable cell count. And you want to use the entire surface, and you can use the volume to figure out what the concentration is and how many you have living in your culture. And obviously you can't count dead cells if you only count things that are growing. Alright, the basic procedure is you have a bunch of serial dilutions that you plate, 20 to 200. You want to go by one order of magnitude each time you dilute. Sometimes you can get a false reading because in the pour plate method you can kill your bacteria by heating them too much. Maybe we'll get to heat sterilization today. Viable cell counts. The plate counts can be highly unreliable when used to assess total cell numbers in natural samples. The idea being that if you take something like a soil sample and then you try and grow that on an agar, it's not the appropriate conditions. It's not the normal environment of the cell, so it can't thrive. The great plate anomaly is that direct microscopes or microscopic countings will always find a higher number than viable cell counts will. And it's not really sure why that happens. Only useful for hand-built. So the pour plate is only useful for bacteria that can survive that pour plate process, which makes it not the best ever. So microscopic methods count dead cells, viable methods don't. Different organisms have different requirements for resources, so you get way different cell counts on things. Turbidity. This is probably more accepted. It's rapid, it's useful, it counts the cells that are in something. You just have a spectrophotometer, you get the optical density, and then you can directly correlate that with growth, because your cells are what's blocking the light as it moves through. So this is showing the machine. Yeah, it's not showing the super great. It's just showing that light gets blocked. That's interpreted as a certain OD. And so the more cells, the more light blocked. So it can be used to figure out pretty much what your cell number is. So you don't want to use wavelengths of light that can be absorbed by the media. So you filter the light, then let it pass through your specimen, and the photodetector will detect it and tell you what your OD is. Next part, turbidity measurements, quick and easy, talked about it. Does not require a significant amount of the sample or destruction of it, and it does not require a lot of time or anything, so it's really convenient. That brings up the fact that it can be problematic. Things that clump or form biofilms cannot use ODs unless you get rid of them, and then they're unnatural, and you have to argue about that. So you have to find your standard curve first. So this is people who've done spectrophotometry. This is what you have to do for that anyway, so it's not anything really different. So this is the growth of two different organisms. The only part you care about is the actual log phase of growth to figure out doubling time. And so you can figure out the doubling time from these curves. Alright, next part. This is Thermus aquaticus. Was found in geysers. They have a bunch of adaptations of their enzymes and lipids and carbohydrates to survive in high temperatures. So we stole their polymerase to do molecular biology in polymerase chain reaction, also known as PCR. Arguably the most important technique in all of molecular biology. So they are an archaea that live in this extreme environment. Taq polymerase is used in it, and PCR can be used for diagnostics, for creating transgenic, like GMO foods, like BT corn. This expresses a bacterial gene that protects it from worms and other parasites. The question with GMOs is whether they cause ecological damage, and so now farmers are required to have a certain amount of non-GMO plants, since we need to keep the biosphere intact. At least in the US, that's the law. DNA constructs can be made from PCR, and these are useful for research and for development of things like DNA vaccines or some biotech companies. Sequencing, so it's sequencing the human genome and everything else, and it's super useful. Paternity test, that's the old school Mare, you are the father. PCR works for basic parts: denaturation, annealing of the primers, elongation, and then repeat the cycle until heat inactivation of the enzyme. Heat, sometimes heat inactivation, sometimes other stuff. Because obviously thermococcus is Taq is able to work in heat. So it might be, I know it goes to 4 degrees at the end, but I think it might temperature lift at the end. This is all I need to know. Those 3 basic steps. T. aquaticus have more saturated fatty acids and more GC content in their DNA, so that they can keep their DNA binded tighter, since GC connections have 3 hydrogen bonds instead of just the 2 of A and T. And these saturated, which just ignore the fact that it's spelled super wrong, fatty acids allow the membrane to not freeze, or not be disrupted. Fever is a critical component in infection because white blood cells have enzymes that work optimally at elevated temperatures. And this is actually control in your immune system. So we're moving away from T. aquaticus and now into your own body. And you don't want your white blood cells always expressing like they're kill the bacteria protein while they're just searching around in your tissues. So only when they get the generalized signal of the fever temperature will they turn those on, basically. They also, the increased temperature of a fever can also be bacteriostatic and keep them from growing. What do we need? What do we need for cell cycle, mitosis, and S phase? S phase, we need helicase, primase, topoisomerase, my favorite enzyme, and single-stranded DNA binding proteins. Obviously, you need DNA polymerase for that. I'm not sure why he skipped it. I think that's supposed to be T. aquaticus. Temperature affects microbial growth. Obviously, E. coli grows at body temperature. Bacillus subtilis does not, probably isn't in the soil, etc., etc. Cardinal temperatures, the minimum, optimum, and maximum temperatures at which an organism grows. So there's minimum, a cold, maximum, the hot, and then the optimum, somewhere between the two. This is for T. aquaticus. Wasn't quite done. This is a mesophile for temperature, and it will grow optimally at a high temperature. Then, if you get much hotter than that, it will start killing the microbe off. Mesophile means the middle. You can have extremophiles and hyper-extremophiles and other categories. Speaking of that, mesophile mid-range temperature, that would be us, 37 degrees Celsius. Psychrophile is something that likes cold things. That's things that grow in the Arctic and in your fridge. Thermophile and hyperthermophile, I'm not sure what the exact difference is between those two. I just know this one's higher. I assume it's like near boiling and above boiling, something like that. Yep, psychrophile goes in the fridge. This is for later, but pasteurization is made to only kill pathogenic bacteria, not all bacteria. After curdling, milk will turn green because Pseudomonas will grow in it, and currently Pseudomonas is my nemesis because it keeps being the right answer. I don't think it is. Hyperthermophile is above 100 degrees C, so I was right, above boiling. You can just logic that, and if mesophile is around human body temperature, psychrophile is fridge, then all that's left, that's a physically significant range, is boiling and above boiling. So here we see the thermophile is basically everything 40 degrees and above, 60 being the primary one. These are all Celsius. Where the hypothermophiles are near boiling or above boiling. Mesophiles like us, around that 37 degree cluster, 39 degrees. That's interesting that that's the maximum. And psychrophile 4 degrees. Know these categories. So psychrophile is the cold one. I think that's the only one that's hard to remember. Mesophile is the middle, because meso-middle. Thermophile is a heat lover, and hyperthermophile is a boil one. Mesophiles love their mid-range temperature, found in warm-blooded animals, terrestrial and aquatic environments, temperate and tropical latitudes. Most important thing, pathogenic microbes that inhabit people are all mesophiles, because they're adapted to the temperature of people. So, warm-blooded animals, terrestrial and aquatic environments, and both temperate and tropical latitudes for these. Microbial growth at cold temperatures. Extremophiles will grow optimally either way, hot or cold. Psychrophiles are the cold, and hyperthermophiles are the really hot. Thermophiles are the hot. We just talked about that. Psychrotolerant can grow at cold, but usually have a mesophile optimum range. And obviously, since they can grow at more ranges, they physically grow in more places. The Antarctic has microbes. Effects of temperature on growth. So things that love the cold, Philae meaning love, psychro meaning cold, production of enzymes that function optimally in the cold. They have some parts of their makeup that make them better suited for this, so alpha helices, a greater number of alpha helices than beta sheets will make these proteins have more actual flex, so if they get frozen they can flex a little bit instead of breaking. More polar and less hydrophobic amino acids, so water is more readily accessible and things are more soluble. They have fewer weak bonds, instead they rely on stronger bonds, and decreased interaction between protein domains. So try and have more autonomous proteins, probably. Transport processes function optimally at low temperatures due to modifications of cytoplasmic membranes, so the high unsaturated fatty acid content. I'm not sure why that's an advantage, but it is. So they inhibit gelling, though. So basically it stops it from solidifying, which makes sense. I assume cholesterol is involved in that somehow as well. So they have. So back to this. Alpha helices. Alpha helices, polar, fewer weak bonds, and decreased interactions between domains. So APFD, a police force detective. Alpha helices, increased alpha helices, a police polar, so more polar. So a police alpha helices, more polar force, fewer weak bonds. So alpha helices, A police force fewer weak bonds. A police polar. Polar. P stands for polar. Alright. A police force detective. Alpha helices, more polar and less hydrophobic regions, force fewer weak bonds, and detective decreased interactions between domains, between protein domains. So, A police force detective. Alpha helices are up, beta barrels are down for flexibility reasons. A police more polar and less hydrophobic. A police force fewer weak bonds. Detective decreased interactions between domains. Microbial growth at high temperatures above 65 degrees Celsius. Only prokaryotic life really exists at all as far as we know. Thermophiles love growing 45 degrees to 80 degrees. And then above 80, so I guess 80 is the cutoff, not 100, but 100 is a convenient cutoff to remember. Inhabit hot environments, and they can be in excess. So these are 80 to probably 120, I want to say that chart said they could go to. High temperatures, hyperthermophiles. In hot springs, they can be chemoorganotrophic and chemolithotrophic. So they take their carbon source from some chemical, and then lithotroph gets it from water. Organotroph gets it from another organism. High prokaryotic diversity of both archaea and bacteria in these high temperatures in hot springs. And then thermal habitats, prokaryotes much more tolerant of high temperatures than eukaryotes. Some of you needing such high temperatures. Organisms with highest temperature are archaea. Not really surprising, archaea tend to be the extremophiles. And non-phototrophic organisms can grow at higher temperatures than phototrophic organisms. Presumably, some of the phototrophic and chlorophyll-like elements can't stand the high temperatures. Growth of hyperthermophiles in boiling water. This is that same thing that Geocorticus. Microbial growth again. So, let's remember the alpha helices, more polar bonds, the fewer weak bonds, and the decreased interaction between domains are first things we know. Some other adaptations: enzymes function at the higher temperature, thermal stability, so critical amino acid substitutions, increased number of ionic bonds between basic and acidic acids, resist unfolding, production of solutes, help stabilize protein, and differences in lipid content. Alright, so we're just going to have to recognize those if they come up. So, amino acid substitutions to have fewer folds, ionic bonds between basic and acidic to prevent unfolding, solutes to help things stay stabilized, and then differences in lipid content. So that's the preventing gelling with more polyunsaturated fats. Microbial growth at lower high pH. pH is a significant effect on whether something can grow. Most are in the 6 to 8 range, like us. That's neutrophiles. They want a neutral pH. An acidophile wants an acidic pH, so a low number, and a kinetophile wants a high pH. Blood is 7.4, and the bicarbonate system keeps it in place. Alright, acidophiles, they love below 6. 6 to 8 is the normal range of neutrophiles, and then lower than that. Acidophiles, there are some obligate acidophiles, and their membranes are actually destroyed at neutral pH. And the membrane is primarily what decides what something can live at. Alkylophiles, they grow best at above 9. I would say above 8, since that's what the previous slide said. And some have a sodium motoforce rather than proton motoforce. so they don't acidify the environment, essentially. So sodium can be used the same way hydrogen is to make energy in the electron transport chain. But it'll look significantly different. The internal pH of a cell must stay relatively close to neutral, even though the external pH is highly acidic or basic. Internal pH has been found as low as 4.6 and high as 9.5 in the extreme cases of acido or alkaphiles, respectively. And microbial culture media typically contains buffer to maintain pH. Also pH indicators, so you know. So sodium chloride, so moving out of the acidified, and the alkylophile range, we are moving into salt. And so you can have a couple of different things. You can have a non-halophile cell, like us, like E. coli, halotolerant. Staph aureus is apparently this, and this is like the psychrotolerant, in that they don't need it, but they have the ability to grow in it. A halophile needs salt. An extreme halophile needs a lot of salt, and that's probably an arcane, I'm guessing. It's an extremophile. Yeah, that's it for those on that page. So osmotic effects. So water's going to go wherever this salt is, basically. So halophiles have a specific requirement for NaCl. Presumably they take in a lot of it so that they can take in water. I think that's what they're trying to say. Extreme halophiles require very high levels of NaCl for growth, and so they're probably using it to underlie some of the mechanisms of growth, which is different from a halophile, which is just using it for water manipulation, basically. Halotolerants that can tolerate the reduction in water activity, but generally grow best in the absence of added solutes. Osmotic effects. Typically, the cytoplasm has a higher solute concentration than the surrounding environment, thus the tendency is for water to move into the cell. That makes sense. Positive water balance. So if it's positive, that means it's moving into the cell. When a cell is an environment with more solute outside, water will flow out unless the cell prevents it somehow. Alright, osmotic effects. Osmophiles. Organisms that live in environments high in sugar as solute. So these are constantly going to have water that moves out. Then xerophiles. Organisms able to grow in very dry environment. Zero being a play on, or I think that's supposed to be pronounced like a Z. I'm not sure. But I'm going to remember it that way because xerophile means they love having zero water. Osmophiles, they like candy, so they like having sugar around them. Honey has a very high osmotic pressure, and that makes it antimicrobial. It can't be contaminated. It never spoils. It is an osmophile stream. Osmotic effects, mechanisms for combating low water activity is by pumping inorganic ions from an environment into the cell to increase its tonicity, and then synthesis of concentration of organic solutes, compatible solutes, compounds used by cell to counteract low water activity in surrounding environment. So basically, you can pump the ions out or into yourself, or you can synthesize a bunch of solutes inside of yourself instead of bringing them in. So the same idea, different mechanism. I'm not going to go through those. Oxygen requirements. Aerobes require it. Anaerobes do not require it, and they may be killed by the exposure. Facultative organisms can live either way, and there's both sides of the fence. There's aerobic facultative organisms and anaerobic facultative organisms. Aerotolerant anaerobes, they can tolerate the presence of oxygen, but they can't use it, and microaerophiles can use very small amounts. And I assume there's anaerobes. It goes like anaerobes, microaerophiles, air tolerant, facultative to aerobe. Alright, so if this is the oxic zone, the anoxic zone, A here, this is clearly an aerobe, this is clearly an anaerobe, this looks facultative to me, this looks like an, no, this, I didn't write what B was, but I'm pretty sure it's an anaerobe. C, this is the facultative one. What is E? This is aerotolerant, so it doesn't really grow differently. There's a facultative, depending on which side it should grow more on one side than the other. And then this, D, is a microaerophile, so a couple can grow. But really, it's just hanging out. I'm guessing this is going to be a question, and so I'm going to go through it once more. This is your aerobe, this is your anaerobe, this is your facultative aerobe in this case. This is your aerotolerant because you only have a couple of colonies. This is your guy that doesn't care which side he's on. So this is actually your aerotolerant. What did I say? This is your micro. Oh, but this is your microaerophile, not your air tolerant. Can use a little bit of oxygen. Okay. Microaerophile, aerotolerant, you would expect to grow the same or go the side. And then microair, you'd expect a couple. Facultative aerobe, you would expect more on one side than the other, but you'd expect to grow everywhere. And then obviously aerobe and anaerobe, quite simple to define from each other. Oxygen requirements. Special techniques are needed to grow aerobic and anaerobic bacteria. Reducing agents are chemicals that are meant to reduce oxygen. So this is not the same as like a normal chemical reducing agent. Or this is like that. Never mind. Don't listen to me, I'm lying. So, reducing agent, trying to remove oxygen from your sample. You can keep something in anoxic conditions. This one in particular is a gel pack that sucks the oxygen out, and then the gel pack itself is a vacuum. So there's 4 electron reductions to get it to be water, and all I should know is that hydrogen peroxide is involved. I'm supposed to remember one of these. Catalase, hydrogen peroxide plus NADH plus H goes to, I want to say, probably 2H2O. Yeah, 2H2O plus NADH. Not NADH, plus NAD plus. So, yeah, it's an important reaction. So, catalase, 1 hydrogen peroxide, 1 NADH to 2 H2O, plus 1H plus goes to 2 H2O plus NAD plus. Staphylococcus aureus, this methicillin-resistant Staphylococcus aureus scare version. It's hemolytic, so it can grow on blood agar. Causes both skin and infection and UTIs. Can also cause toxic blood shock, so it's a real triple threat right there. And it's part of your normal microbiota. It's something like 90% of people will have it.
Its normal form is not bad for us, but it can be an opportunistic infection if your immune system goes down. The important part for me is that it's methicillin resistant. Catalase test is used to find it. That's why I wanted to remember catalase. This is E. coli. It's mutualistic in our guts, forms vitamin K and vitamin something. Gram-negative, cocoa bacilli, generation time of 20 to 30 minutes. It's vitamin B or vitamin K is the other one. Takes up the biological niche in our gut that prevents something pathogenic from being there. Has virulence factor that can cause GI bleeding. It's not the prevalent one in our gut, but we didn't get to talk about those yet. It's an important indicator species. It interacts with an environment and can be sampled to assess the health of that environment. So basically this is, are there feces contaminating your water? So we use it in water contamination to see if it's contaminated or what, and look at it when a public beach or pool gets shut down. This is often what we look at. This is sterilization. Heat sterilization is one kind. There's a couple different kinds. So sterilization kills all viable organisms. Inhibition limits microbial growth. Decontamination makes something safe to handle, and disinfection removes all the pathogens, but not necessarily all microbes. So, in a sort of strange descending order of importance. So, controlling staff, we use sterilization procedures. Water treatment is a form of chemical sterilization. Sterilization can is talked about as killing. Inhibition limits growth. So, like when you put food in the fridge, decontamination makes something safe. Like UV lighting a hood, and disinfection can only destroy the bad pathogenic bacteria, but not all microbes. Most common of all forms is heat sterilization, and it denatures macromolecules. The amount of time to decrease something tenfold, so decrease it to 1/10 of what it was, is the decimal reduction time. But endospores can survive heat sterilization, so that sucks. Autoclave is the most common example. Basically, it uses 120 degree steam and pressure and kills everything. Pasteurization uses more precisely controlled heat to reduce microbial load. So pasteurization is more of not a disinfection procedure. Alright, so that's an autoclave, and anything living except for some extremophiles and viruses should be gone after it's done. Make sure you open up your stuff a little bit before you put it in, otherwise it will explode. And that is not plastic, because it will melt. Decimal reduction time, so this is how long does it take to go down, 1/10. So this is decimal reduction time for this one. This line denotes that, actually, for all of them. And the higher the temperature, the more steep the curve you have. So 50 is not as steep as 60, is not as steep as 70. And the direct relationship between survival and time at certain temperatures is linear. And you can basically calculate it. If you're given a culture of 10,000 cells, number of times reduced to less than a fraction of a cell given a DRT of 10 minutes. So you can basically do this thing where you figure out how long it goes. Autoclave, superheated steam needs to be able to get into the items, will explode due to pressure differential. Make sure plastic that you put in there will not melt. Other heat sterilization, Bunsen burner is one version. There's a bunch of variations of that principle. And so that's for when you need like an inoculation loop or something small. Radiation sterilization, microwaves, your UV, X-ray, gamma, electrons. UV causes double-stranded DNA breaks, and is good for surfaces, but not good for the actual organisms because of those double-stranded breaks. Can't get through any type of light-absorbing surface. So you need an open surface like this in the hood. Radiation is used for sterilization in medical field and food industry. So foods are often irradiated to get rid of microbe contamination. Not going to be a question about this thing, but basically the survival fraction of what you have, and then the amount of radiation you can use to generate this 10% survival curve at each thing. Work out your dosage effect there, or anything on this. Oh, these are all things that are irradiated. Filter sterilization works by essentially having pores that are too small for these organisms to pass it through, but that liquids and gases can go through. A depth filter would be a HEPA filter. HEPA filters trap the microbes and they supply you with oxygen so you don't contaminate your sample. And then a nuclear pore filter works more like a sieve in letting stuff in and not letting stuff in. So this is the nuclear pore working like a sieve. Chemical growth control. There's the three kinds: bacteriostatic, so that's just keeping them from growing more. It's sort of like trapping them in a stationary phase. Then you have the bactericidal, this just kills them, and then bacteriolytic, which kills them specifically by lysing them. And so you would not expect to see any cells after doing that, whereas bactericidal, you would expect to see cells, it would be hard to distinguish living from dead. And bacteriostatic, you would definitely see cells still. Minimum inhibitory concentrations, smallest enough amount needed to inhibit microbial growth. And that will vary with the test organisms used, the inoculum size, temperature, pH, everything about it. Disc diffusion assay is the way that this is tested. Exposed to a certain amount of chemical of interest, so you do a dilution panel and then see what is your minimum inhibitory concentration. How little do you need to have a bacteriostatic effect? How little do you need to have a bactericidal effect, etc., etc. So this is showing what I was saying before, where your viable cell count goes down with bactericidal, but your total cell count stays the same, whereas with bacteriolytic, both will go down because you're literally obliterating the cell wall. If not the cell wall, then the cell membrane or whatever. Chemical antimicrobial agents can be divided into two categories: those used in industrial applications and those in just normal personal human pathogen prevention. And so we can talk about these. These are things like soap and things you use on yourself, like alcohol, to prevent infection. This is industrial stuff that's used, so industrial detergents and alcohol and like chlorine being used to treat water. There's various advantages and disadvantages of the things you have to use. So heat sterilization is fast, but endospores can live through it. And some archaea stuff. Radiation can be dangerous to us. UV is only good for certain surfaces. Filtration only works on fluids, not on solids. And a HEPA filter is an example. Bunsen burner for this. Radiation is UV. Be able to give an example of each one is what I need to do. And decimal reduction time is on the exam figuring it out. So this is the last slide for this. For this test, you can see there's more material, but that's going to be saved for the next test. So basically here, The genome has the entire complement of genetic information for something, includes the genes, regulatory sequences, and non-coding DNA. So this is promoters and stuff, and then the junk DNA that's not really junk, that has some type of regulatory role. In eukaryotes and prokaryotes, it has, usually there's not very much what we'd call junk DNA. Genomics, the category, is the discipline of science of mapping, sequencing, analyzing, and comparing genomes. Alright, this has been my video for the week studying microbes. You can see we went through about 100 slides. If you have any question on this, you can email me at krebradio@gmail.com. If you would like to listen, you can find us on iTunes at KREB Radio. I'm Alex Generous, and good luck on your test and your microbial study.